Okay, welcome back. And we're going to pick up this discussion on epigenetics with a conversation about the microbiome. This is not meant to be an exhaustive source of information. If you're really interested in changes to the microbiome, it's pretty fascinating. There's a lot of literature, really good scholarly scientific literature on the microbiome's influence on the methylation of genes and the expression of genetic traits. So, microbiome, that term refers to all the microorganisms and their genomes present in and on a person. There's a lot of research being done into the specific genome of each microorganism and its impact on the human host. This is considered normal flora. It is non-pathogenic. In fact, it exerts a protective influence against pathogens by taking up space and sometimes by secreting different chemical things that will fight off harmful bacteria. It is unique to every person, and it starts accumulating at birth. So the very first time you put a baby either skin to skin or on an infant warmer, it's picking up bacteria from its environment. That's why skin to skin contact is associated with healthier gut flora. A baby that's placed skin to skin immediately after birth will have gut flora much more similar to its mother than a baby who's placed on a radiant warmer. Those babies, oddly enough, even though we all wash our hands and put on gloves and we observe all of our standard precautions, those babies will actually have flora much more similar to the hospital staff's flora, which is not necessarily a good thing. We get exposed to a lot of superbugs and maybe we're used to them, but a newborn not as much. The first feeding really starts priming that gut for the microbiome. If you feed a baby formula, they will grow different bacteria in their guts than babies who are fed exclusively breast milk. There are prebiotic factors in breast milk that promote the growth of healthy organisms. And these organisms actually exert an influence on things like obesity, diabetes. They actually help that baby express traits that promote normal metabolism and normal glucose metabolism. So that could be really important information for you guys to know if you ever go into maternity. Preserve that patient's microbiome. Antibiotics influence the microbiome. They can destroy healthy flora as well as pathogenic bacteria. And so you're seeing more commonly that physicians will prescribe a probiotic along with an antibiotic to start replacing that normal microbiome. Antibiotic use early in childhood, frequent antibiotic use, those kids that get lots of ear infections and they're on augmentin all winter, those kids have very different microbiomes and they are at higher risk for obesity and diabetes later on in life. Nutrition alters the microbiome. People who eat mostly plants have different bacteria in their guts than people who eat mostly animal-based foods. Processed meat tends to promote the most toxic growth of microorganisms in our gut, the ones that promote unhealthy expression of genetic traits. There's a lot of research being done with obesity and diabetes and the microbiome. There seems to be some implication of Bacteroides and Firmicutes and the balance between those two, and also bifidus bacteria. And at some point in the future, that might become very helpful in preventing illness or treating it. So stuff that you should be aware of. When you're hanging that vancomycin, you should be ordering that patient a yogurt for lunch. Think about it. You want to restore that healthy balance because we don't know yet how many effects down the road that's going to have on them, wiping out all their good bacteria. All right, so getting to epigenetics and cancer. This is also exciting. Cancer originates in changes to both the genome and the epigenome. Changes in the genome, like the Philadelphia chromosome, are associated with cancer. The BRCA gene, we know, is associated with cancer. But what switches those genes on and off is the epigenome. And we know that part of the function of our genes. Some of them act as suppressor genes. We talked about it in the last lecture or 2 lectures ago. And some of them are oncogenes. So think of those genes sort of being like parents. There are 2 functions of a parent. We want to encourage our children to grow and develop. But we also want to stop them from doing all the things they want to do that we know are inappropriate or maybe harmful to them. So we need suppressor genes to provide the discipline, and we need oncogenes to promote healthy growth. If we damage or mutate a suppressor gene, or if we switch it off with a methyl tag or too many methyl tags, we will have inappropriate growth. We will have growth of cells that are less differentiated. They don't want to die. We will not have apoptosis. If we have damage or mutation to an oncogene, it can become less responsive to signals that the suppressor genes are telling it to follow. So any damage to those genes creates havoc in the system. We can have damage to other genes as well. Like I said, the Philadelphia chromosome is a translocation. It's when one piece of a chromosome gets stuck to another in a wrong place, and that encourages the growth of cancerous cells. But really, a lot of it is thought to be managed by these oncogenes and suppressor genes. Methylation is one of the most studied areas when it comes to cancer and the epigenome. Areas around oncogenes are usually hypomethylated. They don't get enough discipline. So they increase their expression and they go, 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 go. It's the parent that encourages somehow the inappropriate behavior, and that inappropriate behavior continues and becomes harder to suppress. Areas around tumor suppressor genes, those discipline genes, are often hypermethylated. This is a parent who's afraid to discipline their child. They don't express that stop message. So they are hypermethylated. They are silenced. Where the oncogenes, the ones that promote things, are increased. Limits are good when it comes to cell function. They need them. So it makes sense that this would inform the research around cancer prevention and cancer treatment. There are under development some compounds to prevent methylation of suppressor genes that would increase their activity, would keep the discipline amongst the cells. There are compounds under investigation that cause oncogenes to be methylated. That means they're not going to promote so much growth. They're going to allow for the natural life cycle of the cell. That cell is going to live its normal life functioning the way it's supposed to. And then at some point after a certain number of cell divisions, it will die. That's what happens when we methylate the oncogenes. Drugs that inhibit enzymes that increase, this is sort of an indirect, it's like a workaround. If we inhibit the enzymes that increase methylation, or if we change how tightly that histone is wrapped, that can also prevent and treat cancer. Antioxidants are under review because they tend to regulate oncogenes, suppressor genes in ways that we don't completely understand. But the basic premise of an antioxidant is that when cells are damaged, there are free oxygen, free radicals that float through the blood. And that can damage cells further. And it can demethylate things that are methylated. So antioxidants like vitamins A, C, and E, resveratrol, phenols, other compounds are under investigation. I have my little chemical symbol here for an antioxidant. Those things can also indirectly influence that methylation process, prevent damage from oxidation, and we'll see what role they play moving forward. I mean, there's a lot of pop culture stuff out there and a lot of marketing around the antioxidants. It's important to find out what works and what doesn't, and what the correct dosage is, and be able to apply those things in the clinical setting. Now we're going to just get into a little debate about nature versus nurture. And we know it's not that simple, because maybe someone else's nature, your grandparents, or someone else's nurture, I should say, maybe your grandparents environment, exposures to DDT, exposures to toxins, exposures to famine, exposures to processed meats, that person's nurture, because of the way methyl tags can be passed on, becomes your nature in a sense. So it's not clear cut, but we do know that environment influences gene expression. There's an interplay there. Environmental toxins. So those of you who are interested in becoming active at the policy level should be interested in this. Things like bisphenol A, BPA. If you've bought a water bottle in the last 5 years, I'm sure you've seen a little symbol that says BPA free. And the reason that that is important is because it has been discovered that bisphenol A, which used to be in every plastic, including pacifiers, bottle nipples, rubber duckies, little vinyl teething rings that we used to give our children, BPAs were in all of those things, and we were ingesting large quantities of them. They are known to be endocrine disruptors, and they disrupt the function of other cells, kidney cells, brain cells. So BPA-free products are a big business. And every once in a while, you will see in the news where a product that's labeled itself BPA-free isn't really. Maybe it's come from a different country where regulations are a little more loose and there's less oversight. But phthalates, they keep plastic soft and supple. They were in everything too. And they tend to disrupt endocrine function. And it's, things like microwaving your stuff in saran wrap or using plastic old Tupperware to reheat things releases those phthalates. And so we're seeing a rise in some diseases that we really didn't see before. Cancers that used to be rare are becoming more common. In little boys, for a while, we were seeing increased rates of hypospadias, which is a defect where the urethra is sort of on the dorsal side of the penis instead of central, where the meatus should be. And that was sort of a kicker that we knew that something was disrupting the normal endocrine processes. Pesticides. We talked about DDT at one point. Pesticides are heavy methylators of certain genes, including your tumor suppressor genes. So it's important to advocate not just for your individual patients and your communities and population health. We need to start thinking about the stuff we put into our environment and how that disrupts our genetic expression. Those things are important. Now a discussion, are identical twins really identical? And I'll tie it around to your practice. It's not just fun facts. Well, their genome is, we know that. They have the same sequence ATCG, right? But their epigenome, not as much. Subtle differences even in utero. Now, as a labor and delivery nurse, I have seen identical twins be born. I will tell you, I have never seen a set that weighed exactly the same at birth. There was always at least a few ounces. Sometimes the differences are striking. For example, when we have twin-to-twin transfusion. So the environment is not exactly equal, even for identical twins in the same amnion, in the same sac, with the same placenta. One twin usually gets a little more blood flow through that placenta. Sometimes that twin gets a lot more blood flow. And we see 1.5 pounds difference in twins. Sometimes it's a matter of where that umbilical cord is. They both have the same environment, but if baby B's umbilical cord gets tied in a knot, or if it gets pinched off in a corner, or it gets compressed during contractions, that baby will get less blood flow. Less blood flow to the baby means that there are stress hormones and catecholamines and other chemical messengers that create like sort of an inflammatory response to help that baby conserve its oxygen. So the environment isn't completely identical, even for identical twins. It is very, very similar in most cases. So the younger identical twins are, the more their epigenome looks alike, and the more alike their genes will act. The more similar will be their gene functioning and their phenotype. So on the left here, can I get my pen out? Nope, I can't. Okay. On the left, we have DNA segments from 3-year-old twins. What they did was they stained the chromatids red and green. Where the red and green overlapped, it fluoresced yellow. Look at all that yellow. It's still not completely yellow. There are still differences even in these 3-year-olds, right? There's still areas that are red and green, but they're more yellow on the left side. That is how their genes are functioning the same. These 50-year-old twins on the right have hardly any yellow. They have lived different lives, and their experiences and their exposures, little differences in the way that they responded to stressors by the release of cortisol, or nutritional choices, or other lifestyle choices, smoking or exercise. All of those things have impacted the way their genes express themselves. And so you see almost no yellow. You see some, but they don't look as identical as these 3-year-olds. If we were to look at newborn chromosomes, we would see mostly yellow, maybe very small areas, which is why you get babies who one twin develops autism and the other does not. So this explains why some twins get a disease and some don't. And what we need to do is study those twins that don't get the disease and prove that the DNA is not our some destiny, and find those factors that are protective against disease. So with that being said, we're going to talk a little bit about the Agouti mice. That was also in the Ghost in Your Genes. Mice with 2 copies of this dominant gene are yellow and obese. And we have always heard if a gene is dominant, it will always express itself. If you have a brown-eyed gene, you'll always have brown eyes. Well, the genes for eye color in a brown-eyed person aren't usually methylated. They're allowed to express themselves. But if you methylate a gene, it doesn't express itself even when it's dominant. So we can do this in mice by methylating that agouti gene, and those mice will have brown coat color and have a normal appetite. They won't get yellow, they won't get obese, and that's what that looks like. So that's sort of a lesson. If we provide nutrients that help methylate certain genes or demethylate certain genes, we can influence the expression of those genes. Dutch hunger winners showed that. It showed that epigenome can pass itself on. When we look at the Swedish county of Norrbotten, that's where we had the maternal grandmothers that experienced famine in their fetal life, and the paternal grandfathers who experienced plenty at the age of 10. And both of those conditions were somehow associated with methyl tags on genes for diabetes and obesity and early death. There seem to be certain developmental windows that those things point to, times when those genes respond to those conditions by tagging them, and times when they're less sensitive and you don't see those changes. And that's been supported in animal studies. There are many animal studies being done on the impact of nutrition on the epigenome and when those things have to occur. Donor nutrients like B vitamins, folic acid, SAM-E, folic acid, when we talk about spina bifida, there's a critical developmental period that if you don't have folic acid, your neural tube will not close. And it is genetically regulated. Some people have a more profound deficiency of folic acid than others, or they're more susceptible to that failure to close the neural tube. So this is the area where nurses can really help by pointing out nutrition. It's funny when we think about saving lives, everybody thinks about those medical doctors who create advancements when dealing with disease and treating disease. Nobody really sings the praises of the March of Dimes nurse who promoted folic acid to clients and reduced the rates of spina bifida. But be an unsung hero anyway. Find out as much as you can about nutrition and epigenetics and use that to help your patients. So how do we use this to improve people's health? How is this applicable to the generalist nurse in practice? Well, getting that family history is important. We need to know what's there. And then we need to know what are the factors that we can modify. We can't modify someone's relatives. We cannot change their genome yet. Someday CRISPR will change that. But even so, that's, extraordinary measures. What we can do is we can tell people what kind of exercise works to turn those genes off. We can tell them how important it is to do that 5 times a week, 30 minutes a day. We can tell them about smoking cessation because we know that that methylates our tumor suppressor genes, and we know that that damages our DNA. We can help people um, eat healthier things to promote a better microbiome. We can teach them about taking a probiotic supplement when they're on antibiotics. There's a lot of ongoing research, a lot of drugs under development that target those methyl tags for application or de-application. A lot of research into the microbiome, a lot of research into what environmental causes make that perfect storm that create disease. And that is the end of our content for this week. Thank you and have a great day.